Cell structure, specifically the cell ultrastructure. We'll start this by looking at a diagram of an animal cell ultrastructure. This is obviously a diagrammatic form, and you need to be able to recognize the organelles in both diagrammatic form and from an electron micrograph. Starting with number 1, this represents a lysosome, the cell terminator that ingests other cell parts that have died or need to be discarded, also involved in phagocytosis. Number 2 represents part of the cell cytoskeleton, not hugely crucial for this part of the specification. 3, 4 and 5 join together to make 6, which of course is the nucleus. Number 3 here is the nuclear membrane. Number 4, the nuclear pore. And number 5, the nucleolus. The nucleolus is where ribosomes are manufactured. Number 7 are the centrioles, which are hugely involved in cell division and are interestingly not present in plant cells. Number 8 is more of the cytoskeleton. Number 9, the mitochondria, the site of respiration. Number 10, the smooth endoplasmic reticulum, or SER, which is the site of lipid synthesis. Number 11 here is pointing at the cell membrane. Number 12, looking at the ribosomes. You find ribosomes both free in the cytoplasm and attached here to number 16, the rough endoplasmic reticulum. Number 13 is the Golgi apparatus. The role of the Golgi apparatus is to package up proteins and lipids, attach carbohydrates to them, and send them off here in what's labelled number 15, vesicles, so they can be sent off to other parts of the cell or indeed to other cells. Number 14 is representing the cytoplasm, which is the site of the cellular reactions. And as we've already said, number 16 is the rough endoplasmic reticulum, which is the site of protein synthesis. Specifically, ribosomes are the site of protein synthesis, but only when they are attached to the endoplasmic reticulum. Now we're going to look at some of the organelles in terms of their electron micrograph structure. This organelle you can identify by the stack of flattened sacs associated with vesicles, and that makes it the Golgi apparatus. This is nicely labelled for you, but if I just enlarge it, it makes it a little easier to see. Here you have the nuclear membrane surrounding the nucleolus. You can't really make out the nuclear pores, but you can just about make out the endoplasmic reticulum. And all of this area in here would be the DNA, or what is technically referred to as chromatin, which is the DNA coupled with the protein. Now, this is, again, a series of flattened sacs, but with very clear black spots, which indicates that it is the rough endoplasmic reticulum. This is usually situated close to the nucleus because the mRNA doesn't need to travel so far from the nucleus then. Now this is the mitochondria. You can see that we have a double membrane bound organelle here. You have the invagination of the internal membrane to form the cristae, which gives a large internal surface area, which enables there to be a large surface area for the reactions of respiration to take place. And last but not least, for this section, we see the centrioles. They are formed by columns of the same molecules which make up the cytoskeleton, and the centrioles are always found at right angles. So if you're looking for them on an electron micrograph, you'll always be looking for a circle and what looks like a rectangle, because they are lying perpendicular to each other, a bit like if you had a pencil. This would be the looking at it long ways, and then this is looking just at the head. It's very important with cell structure that you can try and relate the structure to the function, and that you are completely clear with the names of all of the organelles and what they do, that you can identify both a diagrammatic and an electron micrograph image.